Well, good evening, everyone. Thank you very, very much for coming out at this time of year in particular, and with so many great football matches on just now. Thank you for coming to a science talk. You're very welcome to the University of Edinburgh and to the Playfair Library, which is our pièce de résistance in terms of the architecture. My name's Sue Rigby. I'm Vice Principal for Learning and Teaching at the university, and it's my pleasure to host this event tonight. So this is the Tam Dalyell Prize lecture that we give to someone at the university for their outreach and presentations in science to any variety of people beyond our students. So the presentation that we are going to hear tonight is a huge relief to me, because normally we give the Tam Dalyell Prize to an inspired amateur, a scientist who does a lot of outreach through their own interests. But tonight, it's a delight to give the prize to Sarah Keer-Keer, who is a professional outreach specialist. She works for the university and also for the Wellcome Trust, and she works in an array of settings, taking science to very hard-to-reach communities, taking science to disadvantaged schools, taking science to isolated communities and the Lothians, to places where you wouldn't necessarily expect a biologist to venture. She's one of the co-founders of the Midlothian Science Festival, and her presentations are very widely regarded for both their accessibility and their in-depth analysis of the biology that the Wellcome Trust supports. So the talk tonight is called Life Through a Lens, and I'd very much like to welcome Sarah Keer-Keer.
Hi, and thank you for coming along this evening. I feel very honoured to be given the opportunity to give this lecture. When I found I was the recipient of the prize, I was very pleased, obviously, and I went and had a look at the previous winners and had a bit of a shock to find they were all professors who gave a lecture on their area of scientific specialty. And I haven't done any research for maybe 20 years. I had a moment, a panic. What on earth would I talk about? I haven't, maybe I'm not really a proper scientist. And then I stopped myself and thought, well, maybe I should define who is a real scientist or who is a proper scientist. Can I just begin by asking you that definition? Could you put your hand in the air if you would regard yourself as a scientist? So a small scattering of hands, not too many. I'm hoping to change that. Now, if you didn't put your hand up, could you think about who you do define as being a scientist? And we'll come back to this question as we go through. So Sue Rigby mentioned I work in public engagement, engaging the public with science. And I'm going to talk you through a couple of the things I do, some of my projects. So this is a project we did last year. This is a large inflatable Zorb, which we made into a model of a cell. And we kitted it out. We knitted a full set of human chromosomes to go on the inside. And we put a researcher inside there. And we invited people to come in and experience what it might be like to sit in a cell and to talk to a researcher about cell biology. Another project we do is called Life Through a Lens. And then we use lots of microscopes. A surprising number of people have never used a microscope. And it's a very simple way to engage people. We've used it time and time again and all over the place. So last year we took lots of microscopes to Hong Kong, and the same thing happened. What a brilliant way to start a conversation. Just offer a microscope. It doesn't even matter if someone speaks a different language. They'll always be interested to look at the little things that you're showing them. And it's a good way to start a conversation about science and about, in my case, cells. So as part of that project, we did some art, and it was pretty successful, actually. People were very kind about it, and so I thought, maybe this art thing, maybe there's something in it. And another project we founded off the back of that was a project to use fused glass and other types of glass to communicate really quite complex research science. It's not easy sometimes to get like a complex research microscope out to the public. but I can take these images that are created in glass and recreate them for people. So this is a yeast dividing. I think there's a yeast scientist in the audience. And this is a bacteriophage. But the glass is beautiful and very tactile, and people are drawn to it. People who maybe don't think that they're fascinated by science would be fascinated by this artwork. And it's compelling, draws people over, and it's a great way to start a conversation. These people are in Hong Kong, these people are in Midlothian, they all want to get their hands on the glass. So we mentioned Midlothian Science Festival, of which I'm the co-founder. Now this is a science festival that aims to reach the parts and the audiences that other science festivals can't reach. We don't stay at the university and we don't stay in the city, we go out to community locations and like libraries and community halls and outdoor spaces like play parks and things like that, and engage with people who maybe have never been to a science event. So here's a nice family enjoying some snails in Mayfield Library, and some young rocket scientists also in Mayfield. So I'm really interested in theatre and the way that theatre can bring a story to life. This is a zombie immersive theatre production we did called Edinburgh. And we used this zombie thing that's the zeitgeist at the minute, we use that to talk about how infectious diseases spread and what happens to a cell when it's infected with a virus. Okay, so about 5 years ago, I was asked, well, our centre was asked, really, the research centre, they were asked to put together a project that would talk about our research directly to the public. And we came up with the researchers, they had brilliant ideas, and we created Life Through a Lens, the project I mentioned earlier. And that involved really taking the public through 350 years of history of science and the history of cell biology. And we did some acting. It was fairly terrible, but we did it, and it seemed to work. And we dressed up in period costume, and really, we encouraged a feeling of fun as well as theatre. The Wellcome Trust has an artist who was looking at all these projects, and she created this giant mural, and this is our little piece of history here. This is how our project was commemorated, making science accessible through wigs, because we were wearing so many wigs during the production. So I'm hoping to use that on you tonight, the power of wigs. We're going to see a little bit of that. So stories. Great way to engage people is to use stories. And I'm going to tell you some stories about great scientists, about where science began, how it developed, and where it's going, maybe. So I'll begin with Robert Hooke. You may not have heard of Robert Hooke. Some of you will have. Born in 1635 in the Isle of Wight. He was an adult during the Renaissance time. And some people say he's England's Leonardo da Vinci. He's certainly extremely talented. The man could do pretty much anything. He was a polymath. So, as a child, living in the Isle of Wight, he was looking at fossils down the microscope, for example. One of the first people to record what fossils were like. There's a fossil in this portrait, actually, I think, to commemorate that. Although this portrait wasn't painted during his time, this portrait was painted just a few years ago, because we have no portraits of Hooke, none of his notes, none of his portraits, which is a bit of a mystery, and I will come back to that. So yes, he looked at fossils, but he also was an architect, of course, being a polymath, bit of architecture. He helped rebuild London after the Great Fire of London. He was also an engineer. He did all of the engineering and maths that helped create the dome on St. Paul's Cathedral, although you'll probably think that Sir Christopher Wren built St. Paul's Cathedral. Mr. Hooke was there, but not getting any of the credit. He worked with the chemist Boyle for a long time. Maybe you've heard of Boyle's laws of gases. Well, Hooke helped him develop them, although it's only Boyle we remember for that work. So eventually, Robert Hooke turns his eye to microscopy, and he creates this brilliant book, which is somewhere. Here we go, Micrographia. This isn't a first edition, but there is a first edition in the library in the special collections. It has the most extraordinary plates. So, for instance, this is a fly's eye. I mean, imagine this is created 350 years ago. One of the first people to look down a microscope made this image. It was really extraordinary, and people had never seen anything like it. People really thought that if you couldn't see something, it simply wasn't there. The whole concept of microscopy was not in place in people's minds. There was no idea that there's a hidden world of detail. So, what else did he look at? This is a nettle. Those would be the spines that, would sting you if you touched the nettle. But most horrifying for the general population were his figures of fleas and lice. Because at that time, of course, we were probably all covered in fleas and lice, and we had no idea they had little faces and great detail. And I think people were very shocked. And I think, in fact, Micrographia is the kind of book you got out at a very expensive dinner party to shock your guests. So he was a contemporary of Sir Isaac Newton, and that really is the secret, we think, of why we don't have many records or recollections of Hooke. So Hooke worked on the laws of gravity just in the lead up to Newton taking over and creating his larger theories on laws of gravity. So he was involved, but they clashed over who was, who really did it, I suppose. They clashed that way. And so when Robert Hooke was the first Secretary of the Royal Society, and when he died, Sir Isaac Newton became the second Secretary of the Royal Society. And we think one of the first things that he did was to destroy all of Hooke's notes and all of his portraits, and any written evidence that he'd ever existed. I think that Newton understood about legacy, and he was determined that we would remember his name and not Hooke's. And I think you'll agree he was probably very successful. But one part of Hooke's legacy that does remain in place, I mean, there's several parts, but I'll tell you just about one. So he looked at cork down the microscope, and this is pretty much what he saw. Small compartments, all lined up in rows. And being also the polymath, the architect, this is his, he compared it to the plan of a building with rooms, small rooms in little rows. And this is a monastery, and this is the plan that he compared it to. And of course, the rooms in those monasteries, they're called cells. And he took that word for a room, an architectural term, and he brought it into science. And we use that every day without having any idea where it came from, but it came from Hooke. So actually, standing up and telling you a story is just one way to get something across to you. But theatre is another way, and I did say I would share with you the power of wigs. So I'd like you to welcome to the stage Anthony van Leeuwenhoek.
Good evening, gentle ladies and gentlemen. I am Anthony van Leeuwenhoek, a humble linen merchant. I am from Delft, a great city in Netherlands, alive with new ideas, full of fine merchandise. In my work, buying and selling linen, I must be very sure of the quality of the linen. So I use this single magnifying glass, this lens, to judge the quality of the linen and the fine weave of the cloth. I became very interested in these lenses. I taught myself how to make them. Grinding carefully, sometimes weeks after weeks. I began to wonder, what if I make the lens very small, but much more highly curved? Will I obtain a greater magnification? Huh? Mein Himmel, what a joy that should be. So I worked hard, grinding carefully, and yeah, I obtained the greater magnification that I am hoping for. And still I've worked until I am using magnifications that have never been achieved before. So, now swiftly I turned my attention to looking for things to see in unusual places such as ditch water. And yeah, I see wonders, tiny animals, animalcules I call them. Their motion so swift, so various. They are dancing, pirouetting. They are swimming like eels. So, it was very wonderful to see it. I fall in love with this minute world. I feel a duty to write down my observations to educate those desirous of learning. I decided to write a letter to the Royal Society to tell them of the wonders that could be seen in a single drop of water. And this reply I received. The Royal Society is very grateful to Mr. Leeuwenhoek for his fantastical descriptions. Our greatest experts have never seen anything like them. However, you may continue if you wish to send us your incredible observations. But now I am finding new happy hunting grounds for my molecules. The dirty mouths of not so clean people, beggars on the street, old men who have not washed their mouths all their lives. Yes, and most times I am seeing many very little living animalcules, very prettily moving. And the greater sort have a movement so strong, so swift, they are shooting through the water, or spittle. Like pike move through the water. The lesser sort, they are almost always spinning like a top, and they are much more in number. Since the Royal Society invite, I continued to tell them of my observations. And what replies did I receive? These people in the Royal Society think that I am telling them fairy tales. But I continue to write to them of my observations in many, many letters. For these things I can clearly see. And one day, so shall they.
So the Leeuwenhoek's flamboyant appeals to the Royal Society fell on deaf ears. No one else had seen what he was seeing, and they simply didn't believe him. And people did what even scientists do best, and they ignored him. And when he persisted, they wrote him off as a crank. People who have ideas that are ahead of their time or that don't fit are often in a vulnerable position, and Van Leeuwenhoek was. But he still persisted. And after his 12th letter to the Royal Society, they decided to send a delegation to Delft to see if they could see what he could see. They wanted to see the evidence at last. And he was vindicated because they did see the tiny animalcules moving just as he described. And now he goes down in history as the father of microscopy. The next story revolves around a small but important discovery by the Edinburgh botanist Robert Brown. He was working at Kew and he was working on orchids. And he was looking at the orchid cells. Now, these are the cells from an orchid petal in my office, actually. I made these slides earlier in the week. And he saw just what I saw, which was a spot in every cell, a dense spot. And it made him wonder. And he looked at the leaves and he saw the same thing. A dense, dark spot. And he began to wonder some more. And then he looked at other plants, and he found the same thing. Everywhere he looked, he saw this dense, dark spot. What could it be? Well, when you came in today, you would have found a piece of paper on your chair, and I want you to turn your attention to that paper just now, because this is a chance for you to participate. A really good way to engage you, in fact. I want you to think of the name. What would you call that dense, dark spot? And if you write that name on the paper for me, maybe it's something that's suggested by the size and shape of the spot, or, yeah, I don't know, something about it. Okay, once you've named your dots, could you fold your paper so that you might be able to launch it towards me? Oh. So we want to make these ideas fly. Any kind of shape that you feel might fly or be hurled efficiently in the direction of a scientist. Okay, now just this side of the room first. You'll get your chance later, don't worry. But just this side of the room, could you launch I should have like a big goggles on or something, some kind of health and safety. But anyway, could you launch your ideas to me? Wait for it on the count of 3. So 1, 2, 3. Ah! Okay, I definitely got some. Keep going if you've still got them. I don't want you to miss out. Oh, this one's good. Okay, let's read some of these ideas out. A blood orange, someone said. A sunspot, someone else has said. I like this. Hold on, I'm going to have to put a few things down to carry this on. Putationist, Latin for spot. Okay, I'm noticing, though, that I'm really only getting some ideas from the front 4 or 5 rows and almost nothing from the back, which might be really frustrating if you were sitting at the back and you didn't get it. Reindeer. Flavio. Oh, I like these ideas. Hold on. So this activity is actually a bit of a cheat. It's kind of a way of me talking to you about what happens if you stay in your university campus and ask for ideas to come to you. If you stay there, you'll only get ideas from the people who are immediately already engaged with you. And you'll get great ideas and you'll feel great. And the people at the front will feel great, but the people at the back will get frustrated and they won't have a voice. So I'm going to do something different for the other side of the room. Oh, an eye. I like that one. Yeah, these are all relate to the form of it. Okay, down this side of the room, I would like you to, I'm going to walk out of my safe place and off the campus, and this is your chance to throw this towards me as I move down the room, basically. So that now's your time. Thank you. Ah! I better start reading some out. Ah! Brown's beauty spot. I like that. Oh, sorry. The dot. What's this? Hadvix. Blobosome. So you notice that by just leaving my place of safety and going out into the community of my audience, I'm receiving many more ideas. And no one's quite as frustrated on this side of the room as I think they might have been on that side of the room. And that's a lesson for us all in how to get out of the university and share ideas and share the knowledge and power that we have with other people. Okay, so one of the ideas, well, actually lots of them, came up with one particular name. A lot of people came up with the name nucleus. Does anyone know what that's a Latin term. Does anyone know what it's Latin for? Yes? perfect. It's the diminutive of nux or nut or kernel. That is such a perfect explanation. And I think it was because he used that one because it looks a little bit like a kernel or a nut. So this is the term, not coincidentally, that Robert Brown chose for this nucleus, that for this spot. He called it the nucleus, and he put that idea into a paper, published it in 1831, and presented it to the Linnaean Society. Now, every great theory in science leads usually to new ideas and new theories. It's a process, it builds. And we're going to go now to Berlin in 1837, and we're going to meet two scientists who are dining out together. One is Matthias Schleiden, and he's a botanist who has read that paper by Brown. And another is an animal physiologist who's called Theodor Schwann, and he's been looking at animal tissue. But at this time, we still have no idea what animal tissue is composed of. The tiny dots that we see are called globules now. We don't really have a better definition, and we don't know how they work. So we're now going to see a short play called The Importance of Lunch.
How about this table here?
I think I prefer this table.
Okay. This certainly looks like a nice place.
You really think so?
How was your lunch, Theodore?
Oh, reasonably pleasant, but not very nutritious, I think. And yours?
It was good. About as good as German food gets, really. So, how goes your studies?
Very well, I think. I am helping Müller in his work on digestion. I have a new, powerful microscope. I am seeing animal tissue and globules so clearly. I think I am very near understanding how the body digests food.
A perfect excuse to eat.
Always thinking with your stomach, Matthias. How about your work?
Also good. Did I tell you about this paper by Robert Brown that I read recently?
No.
Every plant cell that he looks at seems to have a dense dark dot in the middle of it. I'm fascinated by the idea. Every plant cell that I look at has this dot, or nucleus, as Robert Brown calls it. What could this thing be doing? What is its purpose?
But Matthias, I am also seeing a kind of dot in the middle of the animal globules I am looking at.
Are you sure?
Yes. Now, certainly, if I stain them, they are more visible. But I myself and Müller have seen a dot in the middle of the globule in the tissue of the spinal cord of chick embryos. Now, what if it is linked in some way to the nucleus you speak of?
But linked in what way?
But perhaps the dot in the animal globules I'm looking at, I'm seeing, is in fact a nucleus. And if they both have the same elementary particle, perhaps the animal globules and the plant cells are more like each other than we have thought. Perhaps, in fact, the globule is a type of cell, which would mean that this cell is the basis of the animals and of the plants.
But Theodore, that would change everything.
Yes.
Sorry, did you want any of those grapes?
So we've met some of the key characters in the history of science, and their theories went on to inform everything that came after. But I'm quite interested in their characters. What characteristics did those people have? What made them great? How were they thinking? Could anyone volunteer like some ideas about how those people approached things that was different or that maybe contributed to their greatness? Shout them out. Curiosity, fantastic. Yes, they stayed curious. That row over there that has these people sitting in it, they were really inquisitive and questioning. So I'm going to think about the key characters, and curious is my first one. Thank you, Shona. What else? Questioning, definitely. Making links across disciplines, so the importance of lunch. So Theodore Schwann could never have had his eureka moment if he hadn't gone out to lunch with a botanist. You have to stay open to talking to people who, from other specialties or people who have different ideas to you. Being willing to learn new theories, even if they conflict with the ones that you're already very fond of. And be prepared to be awkward. I think Tam can relate to this. You don't always say what people want to hear. If you're going to think an original thought, you're going to have to say something that people might not want to hear. So I'm going to talk now about a few of the less famous scientists from the last 100 years. This is William Valentine. This is my grandfather. He worked in a pathology lab. This is him in 1938. I think he strikes a smashing pose. And he won a prize, actually. This is his medal. He won the Sims Woodhead Memorial Medal for services to laboratory sciences. And he was very successful in his field. But one of the stories he told me are the great tips for getting ahead in science, because he did inspire me to, or one of the people that inspired me to study science. He said the thing that had helped him most in his career with promotion and being taken seriously was premature baldness and a fantastic moustache. And I found this just doesn't help women much in science. It's just the same rules don't apply. So he had 2 daughters, and the little baby here is my mum, and he encouraged them to stay curious, and he taught them how to use a microscope. And as they grew up, neither of them became traditional scientists, but they did stay curious. And this is my mum now. She grew up and had 3 children, and the chubby baby this time is me. And all 3 of us were encouraged to be as curious as we wanted to be. We were given a lot of freedom to explore and discover. This is me out and about, amateur botanist. We had a Gerald Durrell type childhood with more pets than was probably sensible. So there's a rabbit under that basket somewhere. And that's the couch is covered in gerbils. And we spent a lot of time in ponds and in rivers, just the freedom to explore, basically. And I can draw a line between these experiences. So I remember the day I watched the crab in the sink for the whole day. And I don't think it's necessarily a coincidence that I ended up studying marine biology. There's a line between our early childhood experiences and what we feel is interesting later on. These were my pet battery chickens that I tried to rehabilitate for several years. And of course, the research I did was about how to improve the housing of battery hens. Again, another line between what I was allowed to do as a child and what I went on to do as a researcher. So my mum taught me how to use a microscope, and my favorite slides in the box were the flea and the louse, just like Hooke. I don't think these things get any less fascinating with age. So I hope I've passed these gifts on to my own children. Oh, I mustn't forget my dad. He's in the room. He wouldn't forgive me. What did he contribute to all of this? He's a lawyer, and he taught me how to argue my corner and some really good critical thinking skills. And it also quite encouraged me to be awkward on occasion. And that's not an easy thing for a girl. I've got to say, this is one of the skills that girls don't necessarily come with, how to stand up and say what isn't expected. So traditionally, we think of science as being taught in schools and learned from books, but my personal experience is that it begins informally. It begins at home with children being encouraged to be curious and questioning and having their questions answered. So here, and it's not just it works with it works with non-scientists and everyone. But I would like to think about what encourages scientific generally, with the public, with children, and with teenagers. I feel like there are some general concepts, and they're not so different from other concepts we've been thinking of already. Curiosity, looking under stones, being allowed to make discoveries. But questioning, why is it like that? Encouraging people to question things and giving them satisfactory answers and finding out answers if you don't know them. Making links. I think that's easier for people who don't have a specialty. Skeptical. This is really something that kicks in the teenage years, but I feel like we should teach all our children as teenagers, particularly, to always ask for the evidence of claims. Learning new information and ideas and theories, and being original and not afraid to say awkward things and things that people don't want to hear. So what are scientists like today? They're really not like they were 300 years ago. They are not a small group of European men. They are women and men, maybe millions of them around the world. And they... in recent years, we've had wonderful science festivals or workshops and things that we can attend that encourage our growing interest in science, even if we haven't studied it before. So I've been impressed with projects like the Citizen Science projects, which are coming up. This is a project in California where you can, as a member of the public, do your own supervised genetic engineering. And this is a project in Japan put together by non-specialists to put Geiger counters in the hands of the public so that they could collect data on radiation after the Fukushima nuclear situation. There weren't really enough of the government around to take this many readings. The public carried these Geiger counters around and they pooled the data, and they could give real-time data to Japanese residents about what the radiation levels were in and around the Fukushima area. So, who are today's scientists? I've got a live link to who I think are some of the more inspiring scientists. A bit of technical wizardry will hopefully link you up soon to some scientists right now. So could you put your hands in the air for a minute? So these people in this room, you are all scientists. If you've come to this event, you're curious to learn new information, and you're questioning, you're beginning to, you are thinking like scientists. Maybe you're the new scientist. So put your hands up one more time, and then put them down again if you think you're not a scientist. Okay, I've definitely convinced a whole raft of the room. I feel like my job here has been worthwhile. Okay, that's really the end of my talk. But there's just a chance for me to thank the Wellcome Trust Centre for Cell Biology, where I work, and the University of Edinburgh for allowing me to be so experimental with my ways that I communicate with the public, and also to all the researchers I work with, without whom I wouldn't be able to do any of these projects. They make it all possible. Okay, so that's all my lovely researchers. Thank you very much. That's the end.
Thank you very much, Sarah. It's very rare that I get to go to a talk where you can throw paper at the presenter and not get shouted at. So that's been a real delight, and I'm delighted that we're all now scientists together. I want to introduce you, though he needs no introduction, to Dr. Tam Dalyell. He was, for more years than is reasonable, one of our principled Labour politicians. And I think particularly at the moment, as the debates go on in the run-up to the election. It's a delight to welcome someone of such integrity and authority. But he's equally well known, probably to most of you in the audience, for his science journalism over very many years. And it's Tam's generosity and originality that has generated this prize that allows us once a year to support and celebrate the science outreach that we do at the university. And we're delighted in the long interaction that Tam has had with the University of Edinburgh. So I'll introduce Tam now and allow him to give the prize to Sarah.
Sue, I'd better confess that when you sent me this year's papers for the prize, that I did raise an eyebrow about the award to Sarah. Because as she said at the beginning, she wasn't exactly one of the professors of science who previously have been the recipient of the university's reward. But I should have known better. Because she was endorsed by the Wellcome Trust, and we've just heard a very original and a very interesting presentation, I shouldn't have doubted the endorsement of the Wellcome Trust, and particularly her boss, David Doherty, who I was on the telephone to this morning, and who gave you a strong imprimatur. It was a very, very interesting presentation. Sue, I don't know whether it is the tradition to take questions.
We certainly can.
Well, I think that no session is complete without anybody having at least the opportunity to put questions to Sarah, because there are so many questions that could be asked. And Sarah, the one thing I implore of you is that in anything that you do, take into account the bureaucratic and administrative burdens that are now put on teachers. Because we now understand that 4 teachers out of 10 in their first year give up the profession. And anecdotally, talking to young teachers, I'm appalled at the pressures that they are put under in England by Ofsted, but also in Scotland, in having to do a vast amount of paperwork. Now, I got the impression from your talk that you were hands-on and didn't make demands of teachers other than what was worthwhile doing at the sharp end. I think what you do is enormously worthwhile, and it's open now for any questions that you care to put.
Thank you. So does anyone have any questions for Sarah?
Make some up?
There must be at least one plant in the audience.
Oh, yes.
Here we are. And are you related to the speaker?
Yes, I'm my mum. Hold on, wait for the microphone.
Would you add imagination to the qualities scientists must have? Is it not so?
So the question is, would I add imagination to the qualities that great scientists should have? I think that that scientists don't always feel like they have a great imagination, but my experience has been that if you encourage their imagination, it grows. They're very creative. I think the glass that you saw at the back is mostly created by scientists, so they're highly creative people. But I think if you want to be truly great, you have to have a wonderful imagination. I mean, how did Theodore Schwann have that moment of realization without a very good imagination. You have to have an imagination to have an original idea.
Well, I'm sure that Mr. Hooke, who came on stage, had a great imagination.
Yeah.
And Newton, too.
It's been obvious to me over my adulthood that scientists have become more and more concentrating on one subject and very fine limits to their investigations. You used the word earlier about Hooke, that he was a polymath.
Yes.
Are we ever going to get back to the stage where our scientists have a broader outlook than the speciality that they've been taught?
Oh, I don't know if I have a good answer to that. You're absolutely right that specialism is how it happens. You can't succeed without extreme specialism. But people who are very good also manage to maintain other interests, and I think, you know, maybe they also play music. I know that one of the PIs in my centre also still can play Rachmaninoff, which I'm deeply impressed by. And I think maybe they can maintain some balance that way, but whether or not you can still be a polymath and a successful scientist, I don't know. I hope so.
Yes, I'd like to answer this.
You want the mic? Here you go.
To my first-hand knowledge, there were two very great productive laboratories. One here in Edinburgh, around the late Ken Murray and Pollock and others. And the second, the Department of Molecular Biology at Hills Road in Cambridge. They had one thing in common. In Cambridge, every coffee time at 10:30, Mrs. Max Perutz would produce coffee and biscuits so that everybody could meet. Certainly Ken and Maureen Murray encouraged a great deal of social interaction in what has become one of the hugely successful laboratories of Europe. And within science, certainly there was interaction. But let no one say that scientists don't interact with other people. I'm amazed at some of those young students who appear on University Challenge, studying chemistry, but how much they know about other things.
Okay, thank you.
I think there was one more question over here. You can put your hand down there.
Yes.
It's on, I think.
I come from Jamaica, and my university, University of the West Indies, has one Nobel Prize winner from about 4 years ago. And I think the key to that was the fact that he was completely open to everybody in the science faculty, no matter what his specialty was. He talked ceaselessly to everybody, picking their brains.
Who are you referring to?
Chen, Professor Chen. Physics.
Yes.
Any last questions?
Well, satisfied them all.
Thank you.
Thank you very much again, everybody. And there's wine at the back if you would grace us with your presence for a bit of a drink.
Could I just add one thing? I'm absolutely amazed at the success of the Science Festival, because I was one of those who was in it at the first year. And do you know how many years I would give it to last? 3 at the most. I'm astonished that it's gone on so long. But that's thanks to many of the interesting lectures that have been produced, and yours is another of them.
Thank you.
Thank you, Sarah.